I'm now going to take you through the AQA GCSE Biology Paper 2 required practicals. And it's all about the ruler drop test, which is how we're going to measure a person's reflexes, so how quickly they can respond to a particular stimulus. So it's a very simple experiment. You need two people. You're going to have one person hold the ruler just above the open hand of another student, as you can see here. Then we're going to drop the ruler, the other student catches the ruler, and obviously the closer that they catch that ruler to the bottom, the faster their reaction time. So I'm going to write that method out for you. So there's my method for required practical 7, the ruler drop test. It is important to be able to react quickly. Many people think that drinking coffee decreases reaction time. Plan an investigation to test the effect of drinking coffee on reaction time. You should include the test for reaction time that you would use and how to make the investigation valid. So I'm hoping this is where you're going to want to write about the ruler drop test. So we're going to describe how we carry out the ruler drop test and then what we're going to do to ensure that the investigation is valid. So carry out a ruler drop test. Let's make sure we give a detail. Make sure you hold the ruler above the thumb. This will ensure that you can actually drop that ruler and catch it in a way that means that a distance can be measured. Our independent variable is what we're going to change. One student will drink coffee, one will not. So that's what we're changing. What about our control variables? It's only worth 4 marks, so you don't need to go too crazy here. So what are we going to keep the same? It's important that you use students of the same age, gender, same amount of sleep, because these will all affect their reaction times. And after all, we're only interested in the effect of coffee on their reaction time. And then we want to repeat and calculate an average. Using a quadrat in order to investigate the distribution of organisms in their habitats. So remember, quadrat is a metal frame that's kind of subdivided. It tends to be 1 meter by 1 meter in terms of its dimensions. And what you're going to do is you're going to place that quadrat randomly using a random number generator to generate some coordinates that will allow you to place that quadrat in a particular field. And then you're going to count the number of organisms belonging to a particular species. So I'll write you a nice straightforward method as to how you're going to use the quadrat in order to investigate the distribution of organisms, and then we'll look at a past paper question. And remember, if you like these sorts of answers, my revision guide is absolutely full of them. Students investigated the populations of earthworms in the soil in 2 different areas. Area A was a grass lawn, Area B was the farmer's field. Chemical X can be mixed with water and poured onto the soil. The mixture brings earthworms to the surface of the soil but does not harm them. Plan an investigation using chemical X to compare the number of earthworms per metre squared in areas A and B. So we're going to obviously need to apply the same concentration and volume of chemical X in order to ensure that we have a fair test, because after all, we only want one independent variable, which is the type of area. So we're going to use our variable layout. After all, it is worth 6 marks. And then don't forget, because we're looking at populations, it's going to be useful to use a quadrat, which is a metal frame, tends to be 1 meter by 1 meter, and we're going to count the number of earthworms that appear. So our independent variable, what we're changing, I will change the type of area we're going to have a grass lawn versus a farmer's field. What is our dependent variable? So what are we measuring? Count the number of worms in both areas present in a 1 meter by 1 meter quadrat. Make sure you're spelling quadrat correctly. Control variables, it is worth 6 marks, so do mention many things that you're keeping the same. So as I've already said, same concentration and volume of chemical X applied to both areas. I've already mentioned the same size of quadrats, so that's also a mark. Same time frame, let's call it 1 hour between application of chemical X and counting the number of worms. And then obviously for reliability, we want to repeat 5 times in each area and calculate an average. And then an extra point to make, because you're using a quadrat, it's very specifically a quadrat point, but you want to place the quadrat randomly in your 2 areas. So you could use a random number generator and plot coordinates. That's what I personally would write to make sure I get those 6 marks.